Thank you everyone for joining us today. I'm excited to talk about our PiggyBac transposon technology from Hera Biolabs. Just an intro to Hera Biolabs. We are a gene editing company. We have two main gene editing technologies. We have our PiggyBac technology, which I'll be talking about today, and then we have our Cas-Clover, which is our nuclease technology, our version of CRISPR-Cas9. And we use this technology in what we call the research field of use. So any sort of research, R&D application of the technology is where we are providing it to interested people. So we use this gene editing technology. We do custom genome engineering, modify cell lines, and create disease models for transgenic animal models. Next we have our SRG rat platform, which we created with this gene editing technology. It's a SCID rat model that we run cancer xenograft services in. And then we've got just a suite of in vivo or in vitro just CRO services. So Hera provides everything from technology licensing through to creating transgenic cell lines, animal models, and then on to providing actual contract services, testing those models in-house. So let me introduce the PiggyBac transposon technology. This technology is sort of the opposite of the mini circles. It's made for stable genomic integrations. So what you do is you transfect your PiggyBac transposon with the PiggyBac transposase enzyme. It basically cuts and pastes the gene of interest, this cargo, into the genomic DNA. And this cutting and pasting transposition activity has a really high efficiency and no real size limit. So this random integration with PiggyBac is located throughout. It's not a specific integration at a specific site. It can be sort of randomly integrated throughout genomic DNA. The main advantages of the PiggyBac technology that we found is that there really is no size limit. So some of the other approaches like lentiviral, you're limited to the size of the genetic cargo you can insert. With PiggyBac, it's been used successfully up to 200 kb and larger. We've optimized the transposase enzyme to be highly efficient. And you also see very high stable expression with the genes of interest that you have stably inserted into the genomic DNA. And so I'll be showing some data basically for each one of these advantages of the technology. The final advantage is down here. It actually can be scarlessly removed. So we have a excision-only version of the PiggyBac transposase enzyme. And you just transfect that and it will actually excise between whatever cargo you have flanked by the ITR sequences and remove it from genomic DNA, leaving back the original sequence. So it's non-mutagenic and you can remove it without leaving behind any sort of scar. So PiggyBac is perfect for large, complex proteins, things that are 10, 15 kb in size or larger. One example would be ion channels or G protein receptors, or proteins or things that have many different subunits or genes that are co-expressed that you all need to be transfected all at once. With PiggyBac, you can actually put them all into one large transposon, or you can deliver them as multiple smaller transposons. Either way works well with this system. So this is one example of a ion channel that was transfected in 3 separate transposons, each about 8 kb in size. And then the stable cell line that was generated after transfection and selection showed the peak current density phenotype that they were looking for, showing that the sodium channel was functional. And then looking at that activity, it was stable expression of the genes and stable functional activity of the protein itself for over 21 passages. And we've seen this time and time again with various proteins, everything from expressing antibodies to basically 4 or 5 different genes for reprogramming. Anything that you'd like to express in a cell, it's highly efficient and super stable when you use PiggyBac transposon system. So here's an example of the long-term stability you get with PiggyBac when you create a reporter cell line. So this group created a reporter cell line in PHP1 cells, which are fairly notorious for silencing transgenes that are inserted. They're very difficult. You have issues with silencing it with most methods that you use with these cells. This group created a reporter cell line. You can see the green fluorescence here is the signal that they were looking for in their reporter. And then 10 months later of continuous passaging without selection, you can see the reporter signal is still as strong and has not been silenced. It's essentially the same as it was from the fresh post-transfection expression of this reporter gene. So yes, it's extremely stable over time. You don't lose expression to epigenetic silencing that makes that can be an issue if you're setting up a platform for screening and you want to have consistency for long term without having any issues or having to go back to the gene editing stage. So PiggyBac is not only highly efficient with cell lines, it's also extremely efficient in making transgenic animal models. This is some example data for using PiggyBac transposon to deliver a bacterial artificial chromosome that was about 300 kb in size, carrying basically the SERP alpha gene, the human SERP alpha gene. And so what this group did is they transfected this back along with the PiggyBac, or not transfected, I guess it was pronuclear injection of the back with the PiggyBac transposase mRNA. And you can see they resulted in pups that are transgenic at a pretty high efficiency. It's not a super high efficiency, but it was successful compared to the direct comparison they did with both CRISPR and TALEN mediated knock-ins at safe harbor sites. They were only able to generate one pup with TALENs with CRISPR-Cas9, and that pup did not go germline. So this is a great way to deliver large cargo into the pronucleus to generate transgenic animal models. Also, the integrations are enzyme mediated, so they're discrete single integrations. They're not concatemerized or chewed up. You have a high percentage of back transgenic animals that actually have the full back, in the original sequence. And that goes for large constructs as well as small constructs. So this data here was published by a group, but it mirrors the data that Hera Biolabs has generated internally over our years creating transgenic mice and rats with this technology. Another example, another route you could do to creating a transgenic animal model would be to modify mouse embryonic stem cells. This group used a 200 kb DNA fragment delivered with PiggyBac. And you can see through fish analysis all the different stable integrations that occurred of this bac in those mouse embryonic stem cells. Because PiggyBac is highly efficient and not prone to silencing, it's a fantastic platform for antibody expression. This technology is being used by lots of different groups to crank out antibodies in large quantities, either for diagnostic purposes, for reagent purposes, or potentially human therapeutic applications. This data is shown here was published by Eli Lilly. They looked at several different monoclonal antibodies and a bispecific antibody, looking at their control, which was their standard method of production of the antibody in CHO cells. And you can see for switching to PiggyBac, the pools of cells had about a 4-fold increase in titer for the amount of protein that was produced, up to 8 grams per liter, which is an extremely high titer for protein production, and about 11-fold increase in the bispecific. They also looked at the stability over time. So the cells were passaged. You can see in each of these, you have generation 0, generation 30, and generation 55. They looked at titer of those cells for each generation as the cells were expanded. And you can see over time, each generation is roughly the same in its ability to produce high quantities of protein. Again, showing long-term stability of transgenes integrated with PiggyBac. And this also saves researchers a lot of time, because whether it's an antibody or whether it's a reporter cell line, you can get robust, consistent expression without having to go through the step of single cell cloning. Pools of cells generally are a quick way to get the answers you need without having to identify a high expressing clone, which can add up to 6 months of time to do the single cell cloning and deliver and screen and then expand and that kind of stuff. So in addition to just being a great platform for stable gene integrations, there's some other applications of PiggyBac that I just wanted to mention. For example, a lot of people are doing genome-wide CRISPR screens. PiggyBac can actually be used to do sort of indel screening as well. So what you do is you transfect a large number of cells and look for basically what genes of interest have been disrupted by integrating the transposon itself. Transposogen, Hera Biolabs' previous name, they got their start creating knockout rats through this method, right? Transposon-mediated mutagenesis of various genes to screen for interesting phenotypes. So PiggyBac, you can actually do sort of loss of function or gain of function. This paper, they did loss of function, right? Dropping in a specific selection marker with mutagenic stop codon exons in both directions. And then they screened looking for cells sensitive to PARP inhibitors, and then looked at where those transposons integrated and had targets that led to PARP inhibitor sensitivity. You can also do, instead of dropping in stop codons, you can also drop in high expressing promoters for gain of function screens, which can't really be done well with other nuclease mediated genome screenings. One way we are using PiggyBac is actually in combination with AAV to deliver the transposon and the transposase in vivo for stable genomic integrations This lets you express your gene of interest in an animal without having to go the route of creating a transgenic, which can take 9 to 12 months to produce a transgenic strain. This lets you co-inject the AAV containing the transposase gene. And then this we did liver specific, but you could do other organs as well. We have a liver specific promoter, gene of interest, GFP, flanked by the ITR delivered. So we co-inject these two AAV viruses, and you can see stable expression in the liver over time. So this method, people do this just with AAV, and you see initial expression, but over time, you lose that stability. It doesn't have the stability because it's not integrated into the genome as it would be with this combination PiggyBac approach. And so this data, we did it with AAV and a nanoparticle encapsulated transposase. And you can see there's high expression and long-term stability with this method for expressing your genes of interest in vivo in the liver of a mouse. You could use this to express a target that you are subsequently targeting with an RNAi therapeutic, or you could just express your specific gene of interest or induce. We've done this to induce various liver diseases or develop or things that would eventually incur cancer development in the liver as well. So overexpressing targets that I think may lead to hepatocellular carcinoma. Yeah, one way we're combining this with our Cas-CLOVER technology is we do a more targeted genomic knock-in with homologous recombination. And then you have the PiggyBac transposon knocked in through homologous recombination with your nuclease, whether Cas-CLOVER or CRISPR or TALEN or whatever nuclease you prefer. And then you have a positive and negative selection cassette that allows you to select for cells with your desired edit. And then you remove the selection marker footprint free, and then you leave behind just your desired gene edit. This would be similar to like a Cre-LoxP system for removing a selection marker, except the advantage is the efficiency for the PiggyBac and the fact that it's footprint free, right? You revert back to original genomic DNA sequence, allowing you to only have your desired edit. A lot of groups are using this successfully in your more difficult to edit cell lines like iPSCs, making small single SNP mutations or things that are low efficiency. This lets you use positive and negative selection to really increase your gene editing success. Another way you can use PiggyBac in general for gene editing is expressing your nuclease. It's, in this case, Cas9 or the Cas-CLOVER mRNA. And rather than transfecting it, you knock in this, the nuclease gene of interest into the cell, not knock in, you deliver it by PiggyBac transposase. Once you have a cell, you can transfect many guide RNAs and see extremely high efficiency in targeting on the various targets, and multiplexing, targeting multiple targets at once. And then once you've successfully edited your cell line and you're done with it, you just pop back out the Cas9 or Cas-CLO51 sequence, and you've got your successfully edited cell line. George Church's lab uses this quite a bit. They've done it to knock out like 60 genes in a pig. And they published some data in iPSCs looking at 42 clones. Of those, you had a high percentage of them either had a knock-in at the target site or that had a knockout. And these numbers show a much increase in the targeting efficiency of nucleases, especially for something like iPSCs.